Hi everyone, my name is Victoria and I am in the Environmental and Life Science program here at Trent, and I'm doing my master's under the supervision of Dr. Barry Seville, and my master's is on Euglena gracilis, or as I like to call it, the microscopic metalhead. So first I want to give you guys an introduction into industrial wastewater. So industrial wastewater is any wastewater or sewage that comes from battery production, the manufacturing of pesticides, and mine runoffs and mine tailing ponds. Now, the issue with these wastewaters is that they often have pollutants in them, such as cadmium, lead, mercury, and arsenic, so these toxic metals that you typically don't want in your water. These metals can have a negative impact not only on human health, but as well as the health of neighboring ecosystems, as these waters can sometimes seep out into neighboring rivers or destroy ecosystems surrounding the industrial wastewater and the mine runoff ponds. Fortunately, we do have some methods to clean up or remove these toxic metals from the industrial waters. So these include your standard filtration system, but it can also include chemical precipitation and resin-based ion exchange. Chemical precipitation just involves using a coagulant to bind the impurities in the water in order to remove them from the water later on. And resin-based ion exchange involves using a resin-based polymer to facilitate ion exchange and swap out the ions and exchange them for another. And this essentially turns a hard water into soft water, similar to water softener. The issue with these methods is that they are relatively expensive and they only remove small amounts of the metal, meaning that they're overall ineffective for a wide-scale environmental cleanup. And chemical precipitation has the added negative effect of potentially introducing chemicals to the environment that wouldn't have otherwise been there, that could potentially lead to other environmental issues. So researchers need to look for a biological organism or a more environmentally friendly way of removing these toxic metals from the environment. This led to the introduction of Euglena. So Euglena is a relatively unique organism. It exhibits traits of both animal and plant cells, and it's found in the aquatic environments of low pH. So these environments are actually similar to mine runoffs, meaning that's already growing in these environments, making it easier to use and implement on a wider scale. Euglena has been identified as a mixotroph, which just means that it can obtain its nutrients one of two ways. It can either obtain nutrients through photosynthesis, similar to plants, or it can obtain its nutrients through external glucose sources in its environment. And Euglena has been identified as an ideal candidate for the removal of heavy metals, specifically because it's been found to produce metal-binding compounds. So I'm going to talk to you about those compounds now. So in 2017-2018, researchers used Euglena biomass, so dried Euglena cells, to bind metals, and they did a chemical analysis, and what they found was that there were actually metal-binding compounds present in the cells. And so they found that these compounds were rich in nitrogen and sulfur, and so they thought that they could potentially alter the sulfur and nitrogen biosynthesis pathways within the cell. And this led to the hypothesis that altering sulfur and nitrogen could actually influence the compounds that are being produced by Euglena. Back in 2000, research was done to see whether or not Euglena was actually able to remove toxic metals. And so researchers in 2000 added mercury into Euglena gracilis cultures and assessed the levels of mercury within not only the growth medium, but within the Euglena cells themselves. So what they found was that over a 6-hour time period, as you can see in figure 1a, that the concentration of mercury within the medium actually declined over the period of 6 hours. And then during that same 6-hour time period, the concentration of mercury actually increased within the Euglena cells, meaning that the Euglena was actually removing mercury from its media and uptaking it into its cells without just seeping it back into the medium. So this was pretty significant. The same group later on in 2003 wanted to see if they could use a pretreatment in order to enhance Euglena's ability to bind cadmium and remove it. So what they did is they pretreated Euglena gracilis cells with mercury before exposing it to cadmium to see what kind of effect this would have. And the results they found were twofold. So what they first found was that it actually increased overall cell viability and the cell's tolerance to cadmium. So figure 2a shows the concentration of living, viable, healthy cells within the culture after being exposed to cadmium, and it compared both pre-treated and non-pre-treated cells. So the pre-treated cells are the gray bars, and as you can see, over a period of time, the only cells that remained viable were the pre-treated cells, the ones that had been pre-treated with mercury. And then figure 2b shows the concentration of cadmium that was actually found in the cells. And what they found was that it also while the mercury pretreatment not only increased the cell's viability and tolerance to cadmium, it also increased its ability to absorb cadmium. So once again, the gray bars are the pre-treated cells and the white are non-pre-treated. As you can see, there is a significant increase in the amount of cadmium that the cell was able to absorb as a result of its pre-treatment. So what this found is that Euglena cells could actually be induced to change, to have not only a higher tolerance, but be more efficient at removing cadmium from its medium. So this was also very significant. This leads me to my research objectives. So my first objective is to pre-treat Euglena gracilis separately with nitrogen and sulfur, and then challenge it with cadmium in order to assess any changes in tolerance as a result of the pre-treatment. My second objective is to identify potential genes that could be encoding for these metal binding compounds using RNA sequencing. So first, I need to figure out whether or not adding extra sulfur or nitrogen to these cells was actually going to cause them to maybe not grow or to change morphologically as a result of exposure to these chemicals. So I started by growing Euglena gracilis in various sulfur concentrations. So this started with the 0 times, which is just the base amount of sulfur that is found in the growth media, all the way up to 20 times, which is a 20 times increase in overall sulfur concentration. And what I found is that even up as high as 20 times, there was no morphological change to the cell to make me believe that there was any negative impact of the sulfur pretreatment. So we went ahead and selected a 10 times sulfur concentration for a sulfur pretreatment of the cells. I performed the same experiment with nitrogen, starting with the 0 base level of nitrogen within the growth media and upped it all the way to 20 times. And what I found was that at 20 times, morphologically, the cell did begin to change. It not only changed shape, but also became a little discolored. It changed from being green and took on a brownish color instead. So this led me to believe that 20 times was going to be a little too severe for what I needed it to do. So we settled on, again, 10 times, as it was high enough to elicit some kind of change, but not high enough for it to really affect the cells negatively. Once I had the concentrations figured out, I needed to figure out long-term if there was going to be any effect on overall growth of my pre-treated cells compared back to my non-pre-treated. So over 66 days, I grew non-pre-treated, sulfur pre-treated, and nitrogen pre-treated Euglena gracilis cells and assessed the overall cell concentration every couple of days. And what I found was that over that period of 66 days, is there was no real difference in growth of my non-pre-treated cells when compared to my pre-treated cells, showing my pretreatment really didn't affect the growth of the cells in any way. Once the pretreatment was picked and completed, I exposed all the cells to cadmium. So I exposed non-pretreated, sulfur pretreated, and nitrogen pretreated cells to cadmium and assessed the concentration of viable living cells that were in the culture. And what I found is that initially there was no real difference in the overall amount of viable cells in the culture as a result between all of my pretreatments as a result of exposure to cadmium. However, by the time you got to 8 days in cadmium, there was a statistically significant increase in the number of viable cells for my cultures that were pretreated when compared back to non-pretreated cultures. What this shows is that the pretreatment actually did have a positive effect on tolerance and did increase Euglena gracilis cells' tolerance to cadmium when compared back to its non-pretreated counterpart. So that was the first objective of my project, and the second objective is to, like I said, identify genes that could be encoding for these metal binding compounds, specifically using RNA sequencing. So before I get into RNA sequencing, I want to talk to you about DNA and RNA. So DNA is double-stranded, and its role is to replicate and store an organism's genetic information. RNA, on the other hand, is single-stranded, and its role is to convert genetic information into a format that can build proteins. So when I think about this, I like to think of a kitchen in a restaurant. So in a kitchen, you're going to have a cookbook, your DNA, because it's the set of instructions for the entire cell or the kitchen. Now, the DNA can undergo transcription, which essentially just means it's making copies. So in this case, this would be the equivalent of photocopying the recipes in your cookbook. And what you're left with at the end, in biological terms, would be mRNA or messenger RNA. But what you're left with in case is the photocopied recipes that are ready to be given to the cook. What I care about mostly is what's happening in this process, is the transcription and mRNA specifically. And so now I want to tell you why we sequence RNA as opposed to doing traditional methods of sequencing DNA. So when we sequence RNA, it allows us to look at multiple genes simultaneously, and it also provides a quantitative result for gene expression. So when you think of gene expression, you kind of need to think of it like a light switch. So a light switch can be turned on or off, but if you have a dimmer on the light switch, you can also change the intensity at which the light's turned on. Gene expression works the same way. A gene can either be turned on or off, but a gene can also be change the intensity at which it's turned on. We call that up or down regulation, so it's either turned on more or less depending on the situation. And RNA sequencing allows us to look at all of these changes within the genes. But before you can get to RNA sequencing, you need to first get yourself some RNA. So I grew up cultures of Euglena gracilis. I grew both pre-treated and non-pre-treated cultures, as well as cultures that had been exposed to cadmium and those that had not, because I wanted a data set that could encompass for all the changes. So to isolate RNA, I used an organic extraction method, which essentially just separated out the RNA from the DNA and the protein and lipids that would also be found. And so once you have isolated RNA, you need to check the quality of it because you want to make sure that you're only sequencing with high quality RNA. This just makes your analysis later on a lot easier and more reliable. And when I say we're checking the quality, we're looking for a couple different things. We want to make sure that our quantity of RNA is good, so we want to make sure we have high concentrations. And we also want to make sure that when we did that organic extraction, we didn't actually take any of the protein or lipids or DNA with it. So we want to make sure that it's pure RNA that we have isolated. And we also want to make sure that it's not degraded in any way or damaged, that we want to make sure it's all intact and good for sequencing. Once you have good RNA, you can proceed forward to the actual sequencing of the RNA. So RNA sequencing typically would work like this. So you would start off with your RNA, and it would get converted through a chemical reaction to complementary DNA. Complementary DNA just means it's DNA from RNA. And once we have the complementary DNA or cDNA, we can actually break it off into fragments and tag it with adapters. And what this allows us to do is it allows us just to know where we're sequencing and what chunks we're sequencing. And then once you have those, what you get back as your data is actually the fragment sequences of your cDNA. So it's the sequences from those fragments. And you can take those sequences and actually map them to a reference genome that you could get off of a database. And this will allow you to look at any changes in gene expression, or we would call them transcript level changes, and see if there's any change in any specific genes or genes all across the board. Now that's, I said, is typical. However, I'm in a position where I have to do a different method because my organism does not have a reference genome. There's no complete reference genome for Euglena gracilis. So I have to do what is known as a Denovo Assembly of the transcriptome. And what this essentially means is that rather than taking those cDNA fragments and aligning them to a reference genome, I need to actually build an RNA framework or assemble a transcriptome from those cDNA fragment sequences. So rather than just mapping them, I actually have to use them to build my framework before I can map them. And then once I have my framework, you're still able to map the reads to that framework in order to identify the changes in transcript levels, identify any changes in gene expressions. Now, while this is more of a nuisance and a bit more time consuming, it does have the added benefit of being able to show us more minute changes in transcript levels or gene expression that you wouldn't otherwise get to see if you had just done the traditional method. So while it is a bit more work, it does have the added benefit of being able to show us those more minute changes. Now, I've kind of talked to you about my project a lot, so I want to bring it back and end it off by explaining why this is so important. So if we can improve our understanding of how Euglena gracilis is able to bind these heavy metals, we can actually potentially use this and harness this ability for real-world applications, such as actually applying it to mine runoffs, applying it to the real industrial wastewaters, and make a difference. Because if we can harness this ability, we can potentially increase the removal of these harmful metals such as mercury, cadmium, and lead. And so we can help make a difference in cleaning up some of the water. And it does have another benefit, not just environmental. So I do mention that it can remove harmful metals such as mercury and cadmium. However, it can also remove economically relevant metals such as gold and silver. And what this could do is actually provide an economic incentive for organizations to want to switch over to this method. So if a mining operation is finding that it's losing some of its yield into the wastewaters as it's flowing out, we can use some of this to reabsorb and remove some of these metals. We can sell it as an economic incentive to actually use this method because you can recoup some of those losses. And even if you can't recoup some of the losses, it can maybe help offset the cost of using this method as a cleanup method. So not only is it environmentally friendly, but economically it also is a viable option, which is not common that you see in environmental cleanup. So it has that added benefit. And I just want to say thank you for listening to my presentation.